Hey everybody, Pre-Med Mama here. Thank you so much for joining me on my channel. Here, we'll study together for the MCAT by reading aloud each chapter and section of the Kaplan MCAT review books, one at a time. Today, we're in Chapter 1 of the Biology review book by Kaplan. In Section 1-5, we're learning about viruses and subviral particles. Today, I'll be reading about the viral life cycle. Thank you again for joining me. I'm so glad you're here. And if you haven't already, please be sure to subscribe to my channel and share with friends and family. You got this. All right, jumping right into the viral life cycle. As we have already discussed, viruses must infect a host cell and use the host cell's machinery in order to reproduce. Here we will discuss the viral life cycle in detail, starting with infection. Viruses can only infect a specific set of cells. In order to infect a cell, the virus has to bind to specific receptors on the host cell. Without the proper receptors, a cell is essentially invisible to a virus. Pretty neat, huh? Once the virus binds to the correct receptor, the virus and the cell are brought into close enough proximity to permit additional interactions. I like to imagine a spaceship and a space shuttle in outer space and how the shuttle has to attach to the receptor on the other ship in order to connect and lock. That's kind of how it works with a cell and a virus in terms of the receptor and the binding interactive process. Enveloped viruses fuse with the plasma membrane of a cell, allowing entry of the virion into the host cell. Sometimes a host cell may misinterpret the binding of a virus to the membrane as nutrients or other useful molecules, and will actually bring the virus into the cytoplasm via endocytosis. Crazy, right? As mentioned earlier, bacteriophages use tail fibers to anchor themselves to the cell membrane and then inject their viral genome into the host bacterium using the tail sheath. Some tail fibers even have enzymatic activity, allowing for both penetration of the cell wall and the formation of pores in the cell membrane. Depending on the virus, different portions of the virion will be inserted into host cells. Enveloped viruses such as HIV fuse with the membrane and enter the cell intact, whereas bacteriophages only insert their genetic material, leaving their capsids outside the host cell. Next, we'll move into translation and progeny assembly. After infection, translation of a viral genetic material must occur in order for the virus to reproduce. This requires translocation, or moving, of the genetic material to the correct location in the cell. DNA viruses must go to the nucleus in order to be transcribed to mRNA. The mRNA, or messenger RNA, then goes to the cytoplasm, where it is translated to proteins. Genetic material from positive sense RNA viruses stays in the cytoplasm, where it is directly translated to protein by host cell ribosomes. Negative sense RNA viruses require synthesis of a complementary RNA strand via RNA replicase, which can then be translated to form proteins. DNA formed through reverse transcription in retroviruses, remember HIV is an example of a retrovirus, also travels to the nucleus, where it can be integrated into the host genome. Using the ribosomes, tRNA, which stands for transfer RNA, amino acids and enzymes of the host cell, the viral RNA is translated into protein. Many of these proteins are structural capsid proteins and allow for the creation of new virions in the cytoplasm of the host cell. Once the viral genome has been replicated, it can be packaged within the capsid. Nice and tidy, right? Just packaged right up. Note that the viral genome must be returned to its original form before packaging. For example, retroviruses must transcribe new copies of their single-stranded RNA from the DNA that entered the host genome. A single virus may create anywhere from hundreds to many thousands of new virions within a single host cell. Wow, that's amazing. I'll be creating another short video immediately following this one with the two other components of the viral life cycle, which are progeny release and the lytic and lysogenic cycles. Check back soon, and thanks for watching and listening.